Hello students, myself Saurabh Yashaswee, Assistant Professor at Department of Zoology, RD and DJ College, Munger University, Munger. So today we will be discussing about a new topic, the neutral theory of evolution. So to start with the evolution, I must say that initially the concept of evolution was given by the Darwin. And what Darwin thought of evolution was that the evolution is a result of interplay between variation and selection. What we mean about this? We mean that in each generation there are vast number of variations that is produced by a species by the mutation of the gene and by random assortment of genes in reproduction. And those genes which give rise to the character that adapt to the environment and make the individual survive to the best are selected. So basically, when we talk about the Darwin theory of evolution, we are talking about the natural selection, and the natural selection defines how fit the survival would be. But after Darwin, there was a period called as Neo-Darwinism, in which different people thought that beyond Darwinism, there are some more things, there are some more principles that can add to Darwin theory. And one during this period, there was the rise of the Mendelian genetics, which was proposed by the Mendel. Then other scientists like Mueller, who described about the gene, Fisher, Halden, and C. Walright, who discussed about the population genetics, and Dobzhansky, who studied natural population studies, and Simpson, who studied the paleontological analysis, and the ecological genetics studied by the Ford. These studies made sure that beyond Darwin, there is some concept that also drives the process of evolution. So these studies built a large and impressive edifice of Neo-Darwinian theory. So people tried to explain everything, but in due course of time, what they told, they talked about the gene pools, they talked about gene frequencies, any population change, and a lot of things about evolution. But these studies were necessarily based on the observation of a phenotypic level. But what actually goes on in evolution at the level of internal structure of gene remained undefined. At this stage, there was a scientist called who was known as Kimura, who proposed the neutral theory of evolution. And in his theory, he proposed that the molecular level is important for evolutionary change. And at this molecular level, this evolutionary change is brought about by the random drift of selectively equivalent mutant genes. He meant that they are adaptively neither more or less advantageous than genes they replace. Means the genes that are replaced in the process of evolution and the genes which are not evolved during the process of evolution both have same adaptability equivalence. To be more clear, it can be clarified that the random drift during the random drift the selectively equivalent mutant genes, they are adaptively neither more or less advantageous than the gene they replace. So let's have a look about what he wanted to say. We would we should look at it in the explanatory form. So when you look into this table, we see there are three columns. The first column is the type of change. The second is human alpha versus human beta, that is the hemoglobin chain of the human. Then carb alpha versus human beta, the hemoglobin chains. So here we are comparing two things. We are comparing the alpha chain of hemoglobin of human to the beta chain of hemoglobin of human. And in second column, we are talking about the alpha chain of the carb hemoglobin versus the beta chain of the human hemoglobin. So when we see the number of amino acids in human alpha beta hemoglobin is 147, and in carb alpha hemoglobin it is 149. So total we are looking on those number of amino acids. So what we find that when we see the in alpha hemoglobin of human and beta hemoglobin of human, when we compare it, there is no change at 62 amino acid position. For 62 amino acids there is no change. Similarly, in case of carb alpha and human beta hemoglobin, the different one, there is no change for 61 amino acids. Means 61 amino acids of alpha hemoglobin chain of carb and human beta hemoglobin chain are same. Similarly, when we look into one nucleotide change, we find that in the right side of the column we have 55, on the left side of column we have 49. For two nucleotide change we have 21 in human versus human alpha and beta chain, but in case of carb alpha and versus human beta we find 29. The gap for alpha human and beta human is 9, and for carb alpha and human beta is 10. So when we see uh, the parameter human beta remains same for both the columns. What we are comparing here is the alpha chain of hemoglobin of human and the alpha hemoglobin of the carb. So the column at the left categorizes the amino acids according to whether there is no change. A change due to minimum of at least one amino acid and at least two substitution in genetic code or a gap. So what we find it here that the number are similar in both the cases, almost similar. I'm not saying exactly the same, I'm saying similar. Whether one compares the chain in the same species, that is human alpha versus human beta, or in two species, that is carb alpha versus human beta, suggesting that alpha chain in two lineage have accumulated mutations at the same rate for 400 million years ago. That means if we are seeing a very less change or we have similar chains, that means that the mutations that took place in alpha hemoglobin of the human or the alpha hemoglobin of the carb, they are almost similar within a certain span of time. So this, in short, we can say that the evolution, uh, what was that in a given protein, the rate at which amino acids are substituted for another is about same in the many diverse lineage. So here it explains. Well, in the last slide we have seen that the alpha chain of hemoglobin and the beta chain of hemoglobin of human compared to the alpha chain of hemoglobin of carb and the beta human chain of hemoglobin of human, both have the similarities, right? So both the species are different, one is human, other is the carb. So what the Kimuras neutral theory, the first salient feature of this theory is that for a given protein, the rate at which amino acids are substituted for one another is about same in many diverse lineage. So the lineage for humans and for the carp is different. But neutral theory says that although the lineages are different, the rate at which amino acids are substituted is the same for a span of time, over a span of time. This is the first thing. So moving on to the next thing, we must look that the findings, the second finding was that the nucleotide substitution in the course of evolution must be result of random fixation of neutral or nearly neutral mutants rather than a result of positive Darwinian selection. So we can say that this neutral theory is anti to the Darwinian selection of natural selection. So you can see a graph where there is a x-axis which shows time span in billion of years ago, as well as y-axis which has nucleotide substitution. Here you can see there are two dots. The one dot is filled and the other is empty. So the dots which are filled, it represents the 7 pair of proteins in 16 pair of mammals, and these dots which are empty, which has no color, belongs to the primates. So what we see here, that except for these primates, the dots which are not colored, we see all those colored dots are the points, or those dots which are the points fall close to the straight line. These are all the colored dots you can see here falls on the straight line, with the exception that belongs to the white dots which is of primates. What it suggests? It suggests that the protein molecular evolution rate is uniform, because you see that here there are 7 pair proteins in 16 pair of mammals. All of them, the colored ones, except for the primates which is not colored, they fall near the straight line. So it means that the protein's molecular evolutionary rate is uniform. And if it is uniform, it deviates from the Darwinian theory of natural selection. So this was the second finding. This was the second important postulate or the finding or the theory of the neutral selection. So when we move to the next slide, we find that how this selection is defined. So we know about the protein polymorphism. So this protein polymorphism is selectively neutral or nearly neutral. So it must maintain in a population the balance between the mutational input and random extinction. So let us see what does it mean. So on the right hand of the table, the column says protein, on the left side is the evolutionary rate. So you look to the protein, this is fibrinopeptides. If you remember this protein remains as a precursor molecule, and it is useful, it gets cleaved during and forms the active protein during the blood clotting. So this fibrinopeptide has the functions after being cleaved in blood clotting. Then you see pancreatic ribonucleases, hemoglobin chains, myoglobins, animal lysosomes, insulin, cytochrome, and histone 4. Histone 4 are said to be highly conserved proteins. And accordingly, you see the evolutionary rate. Fibrinopeptides is 9. That means and the histone is 0.006. And you know how important is a histone. Histone is the basic protein that forms the structure for the DNA, the octamer structures for the DNA. Cytochrome is essentially important. Insulin is essentially important. So the proteins which are very important, which are functionally important, they have a low evolutionary rate. But the proteins which are needed only during a certain time or which have less functions has a high evolutionary rate. So here we say that the rate is given for, here we see that the rate is given for several proteins. The rate is particularly high for fibrinopeptides, which appears to have a very little function, and the rate is very low for those proteins whose precise shape is significant and hence subjects them to the stronger constant of for evolution. So basically, when we summarize it, then we say that proteins which have important functions has low evolutionary rate, and proteins which have which have less important functions have high evolutionary rate. And so there is a balance. The balance, the mutational input, you can see the balance is necessary between the mutational input and the random existence. So this was the third important point for the neutral theory of evolution. Moving on, when we look further, we can come to the conclusion that the neutralist would think that some mutant can be spread throughout the population of their own without having any selective advantage. So if you remember correctly about the Darwinian theory, it always speaks about the advantages of the natural selection. But here, for neutral theory of evolution, the selective advantage is not compulsory. If a mutant is selectively equivalent to the pre-existing allele, its fate is left to chance. That means its fate is left to chance means that if a random genetic drift occurs, it may be, and it persists for a longer period of time, then there is a chance that new species or new population can be formed. So the next thing is that its frequency, the frequency fluctuates, increasing or decreasing over a time because only a relatively small number of gametes are sampled out. Then how does this frequency fluctuates? Let me give one example. Suppose in a population of 10,000, if 10 people die, the percentage is 0.01. But in a population of 100, 10 people die, the percentage is 10%. So you see, in a smaller population, even a small decline can change the frequency to a much higher level. So neutralists here say that there are two things. That first, mutation can spread to a population without having any selective advantage, and they are selectively equivalent to the pre-existing alleles. And the frequency is highly fluctuating, and because of the small number of gametes that are sampled out or a small population size. So here this is the thing. Then moving to my next slide, what it says that in course of random drift, when last slide we talked about the chance and the random drift, the overwhelming majority of mutant alleles are lost, but the remaining minority of them eventually become fixed in a population. As I told earlier, that if a chance occurs, if there is a random drift, then the majority of mutant alleles are lost. But if a small amount of them, or the minority of the mutant alleles, becomes eventually fixed in a population, and if neutral mutations are common at molecular level, and this random drift continues over a long period of time, say millions of generations, not just 1 year or 2 year, millions of generations, or that means million of years, the genetic composition of population seems to change or can change very significantly. So as I said, that the random drift, it occurs, it is because of the small population size, and the majority of the mutants are lost, minority which are persistent, and if random drift continues to continue for a longer generation of time or a longer period of time, then the genetic composition of population seems to be changing significantly, right? So when we look to the next slide, here it comes that for any neutral mutant that appears in a population, the probability of eventual fixation is equals to its initial frequency. What does it mean? So let me explain. Suppose we have a neutral mutation. Neutral mutation means that even if there is a mutation, it has no selective advantage, right? But if it appears in a population, then what is the probability, right? So we talk that if it appears in a population, the probability of eventual fixation, that if it is there, if there is a minor mutant allele and it gets fixed in a population, then its eventual fixation is equals to its initial frequency. The frequency it had during the beginning is its probability. So let us suppose that in a DNA, at a position we have a neutral site at which all alleles are selectively equivalent. Not one has advantage or the other has a disadvantage. Every allele is selectively equivalent. And the rate of mutation per generation is U. So in a haploid population of size n, if we consider a population size n, then NU means the number of the population size into rate of mutation occurs at this site at each generation. And if there is no selection, as neutral theory states, and if there is no selection, all genotypes have same probability of each fixation. So under a neutral model, the probability that an allele or mutation fixes it is simply its relative frequency in the population. Simply, n size population, mutation rate of mutation per generation is mu. So Nu is the mutation rate that occurs at this site at each generation. And as the neutral model says, the probability that allele or mutation fixes is simple its relative frequency in the population. So correct that if the population size is n and there is a mutation occurring, then this is correct. But what happens if there is a new mutation? So let's have a look to that, and we find that if there is a new mutation, the relative frequency changes to 1 by n. So remember in the last slide there was no mutation and the frequency was n into u. For the new mutation, the relative frequency is 1 by n, and thus the probability that new mutation reaches is simply 1 by n. So if we take k as the rate of substitution per generation, rate of substitution of amino acid at a site per generation is obtained simply by multiplying the mutation that occurs at each generation. That means this is Nu. So here you see the equation k equals to Nu into 1 by n, right? So then where k stands for rate of substitution per generation. Rate of substitution per generation, and Nu is, remember in the last slide, Nu is the rate of mutation of one generation, and 1 by n is the frequency of new mutation. So here you say that if we multiply nu into 1 by n, we will get u. That means that the rate of substitution is equals to the initial frequency. Remember, u in last slide was the initial frequency. So this is that the rate of substitution per generation according to neutral theory is the initial mutation rate or the initial frequency. So now moving to the next slide, where we can see that how this um, how this mutation is effective, and how this mutation has affects the population size. So suppose if we talk about mutation, then we can have neutral mutation, we can have deleterious mutation. So it depends upon the fitness effect of the mutation. Suppose the fitness effect of mutation, where we call as the selection coefficient, is S, and we have effective population size of Ne, right? So if Ne into S, the effective is less than 1, the fate of mutation is essentially determined by the genetic drift. And if it is determined by the genetic drift, then it is considered effectively neutral. So in other words, in a small population, the effects of random genetic drift overcomes the effect of selection. So here it proves that the natural selection doesn't stand as an option for neutral theory. For a population that is of effective size Ne, and if the selection coefficient is S, and if Ne into S is less than 1, then the fate of mutation will be determined by random genetic drift and not by the effects of selection. That means all mutations for where Ne into S is less than 1, the mutation is neutral. So when we look to the next slide, we find that how and what is the average length of the time until fixation takes place. So it is 4 times the effective population size, that is 4 into Ne. And by meaning of effective population size, we means that it is the number of breeding individual in one generation, and it is much smaller than the total number of individual in the species. That is why we call it as effective population size. So this is all about the neutral theory of mutation. So let me give you a conclusive remark for this. So when we talk about the concluding remarks, we have that the first thing is that the neutral theory of mutation is just anti, or it defies the natural selection theory. So in this theory, the mutation remains equivalent for all the alleles, and the mutation here, no natural selection works here. So when you study or you look into the selection process, we are looking into the substitution, and when we look into the substitution process, we find that the substitution process is completely neutral. It can be in the population, it can remain in the population without getting extinct. The second thing is the rate of evolution for neutral mutation is prolonged, and those proteins which have great functions, which have some important functions, their evolutionary rate is quite low. But the proteins which have little functions, their evolutionary rate is quite high. Then we have also looked that the lineage, for different species, it is not necessary that substitution may occur very randomly. And moreover, you have seen that the alpha chain of a human hemoglobin compared to beta hemoglobin chain, and the alpha chain of carb hemoglobin when compared to the beta chain of human, they have striking similarities. So these are the important things, and when we look into and these are the very important things which you can look into while studying this neutral theory of uh, evolution. So this is the topic for the day. We stop here, and we will meet again with a new topic. So for uh, uh, so far we have seen it, and we will I will upload this on my YouTube. And thank you for watching. Stay at home, stay safe.